Hi, everyone. So today's going to be my guest lecture. So I'll be lecturing on the DNA of ancient Egypt. So by me. So I think this is going to be a really, really fun presentation. I hope you guys enjoy it. All right, so the goal of this presentation is just to describe the ancient DNA analysis and workflow through the lens of ancient Egypt. So Thais, you have an amazing breakdown on just the wet lab and the dry lab methodologies and like the whole ancient DNA analysis. So this is more of like a case study. So how it can be applied to archaeology or bioarchaeology. So we'll discuss the archaeogenetics of ancient Egypt. We're going to learn how ancient DNA can be modeled. So modeling historical populations and migrations. The best example I can think of is if you guys have ever done 23andMe, do you know how like whenever it gives you the ancestral breakdown? Well, we can actually do that with ancient populations, so not just modern. And then lastly, examine the concept of race and ethnicity in ancient and modern Egypt, because this is a hot topic right here that's been debated for over a century. But before we begin, I want to ask you guys, just think, which ancient civilization do you think is represented the most in the ancient DNA corpus? And a lot of you guys will probably be thinking ancient Egypt, but that is actually not the case. And I'm sure a lot of you guys think ancient Egypt because of mummies and all that. There's hundreds of thousands of mummies. So actually, I think Thais, she showed us a breakdown of where most ancient DNA comes from. So it's very, because I mean, the field began in Europe, so it's very Eurocentric. Most of your samples are from ancient Rome, ancient Greece, not the ancient Middle East. So that's something interesting. But why is that the case? So why don't we have more mummy DNA? Well, the mummification process actually is breaks down DNA. So you have a chemical such as sodium carbonate, also known as natron. It's used in the mummification process. It breaks down DNA. On top of that, the dry desert climate. So you have dry desert climate, the humidity in tombs, because these are enclosed tombs with high humidity. So with all this stuff combined, and on top of that, ancient DNA is already very, very degraded. So add all this stuff and you have little to no DNA, unfortunately. But that being said, we actually do have samples of ancient DNA, like from mummies, and we can actually extract DNA from mummies. It's been done before, and we're going to see later that it's somewhat common. It's just very rare and it has a very low success rate. And also another interesting thing is a lot of the mummies that we have were discovered in the early 20th century or the late 1800s. And contamination was a very big thing. Like as we saw in the previous presentation, whenever you like extract DNA, you have to be in a airlock lab, and it's very, very sensitive to contamination. So you had all these Egyptian scholars, early Egyptology scholars in Egyptology, contaminating the mummies, and because of that, we can't really extract the DNA from those. And a fun story I've always heard was, I think this was for maybe Ramses II. He was the pharaoh of Egypt. They found his mummy, and they were doing like an analysis on his intestines, and they found traces of tobacco. And all the Egyptologists were like, what in the world? This is crazy because tobacco is a New World crop. But it later turned out that it was just contamination from over 100 years ago. Probably some Egyptologists or some scholars smoked tobacco and touched it, and that's tobacco in their intestines. All right, and here's another thing. So we actually do have a lot of mitochondrial DNA of Egyptian mummies, although that is not the DNA we want. So we actually have two different types of DNA, or actually there's more, but these are the main that we use. Nuclear DNA and mitochondrial DNA. So mitochondrial DNA, often you'll see it abbreviated as mtDNA. It's inherited maternally. So yeah, maternally inherited and can only trace maternal lineages. So if I also get my mitochondrial DNA, I could see my mom's. Like I can keep going down until we can't anymore. And it has about 16,500 base pairs, whereas the normal human genome with nuclear DNA has about 3.2 billion base pairs. So this is like really a big difference in the amount of DNA that you can actually get. So early in the field, mitochondrial DNA was actually the first type of DNA we could extract because you have over 1,000 mitochondria in the cell for power, mitochondria powerhouse of the cell. Yeah. So it was much more easier and more efficient to extract. But now, thankfully, due to new DNA sequencing technologies, we can actually get the entire DNA. So this is the nuclear DNA. And it's harder because you only have one nucleus in the cell versus over 1,000 mitochondria. And with nuclear DNA, we can actually compare genetic relations on an individual level. So we can see, for example, my DNA compared to like a large population instead of just tracing maternal lineages. And this is pretty groundbreaking. And here's just a diagram of the different type of DNA. That being said, we actually do have DNA of ancient Egypt, nuclear DNA in ancient Egypt, although we only have five. We have hundreds of mitochondrial, but only five nuclear DNA from mummies. And does anyone know where this picture is from? Anybody? Maybe? Oh, you know, yeah, you know, got the Carlos Museum. Highly recommend it if you guys haven't checked it out. So we have, yeah, five mummies, and I'm going to go through each of these and just explain them all. But now an interesting thing is, so the oldest mummy sample that we have is the Nueret individual, and the newest is the JK288 from the Ptolemaic period, so the Greek, ancient Egypt during the Greek rule. And it's interesting that JK288 is actually closer to us than it is to the oldest sample. So that's just how ancient Egypt is. And I think this kind of goes back to the whole, I don't know if you guys have heard it, Cleopatra is actually closer to our time than she is to the building of the pyramids. So ancient Egypt is very old, so we have a lot of gaps in the DNA record. Awesome. So let's go ahead and go into the first individual. And this one was actually like a groundbreaking paper. This was in 20, like literally less than a year ago. I think it was in Nature, I want to say, or Cell, one of the two. So this is the Nueret individual, and here's a reconstruction of him. They found his full skeleton. He probably lived about 100 years, probably even saw the Great Pyramids being built in his lifetime or begun to be built. And the interesting thing is the reason they were able to preserve his DNA and extract it was because he was not mummified. He was actually interred in a large circular clay pot. So this was like an early pre-mummification burial process in ancient Egypt. He was about 5'2", 5'3", likely died at the age between 44 and 64 years old, although the paper says likely towards the upper end, so probably 50, 60 years old. And this is actually interesting. So they believe that he was a potter because they were able to examine the stress marks in his skeleton, and it showed patterns of osteoarthritis, so which is consistent with that of a potter. And there's actually an entire book on this called the Atlas of Occupational Markers on Human Remains. So you can just look at bones and tell what a person's occupation is based on their maybe stress in certain areas. So I thought that was pretty cool. So this is the Noira individual. And now this actually brings us to the ancient DNA downstream process. So for example, how do you now plot to see like where does this individual come from? How do you like actually do the downstream analysis for their DNA? And this is the first thing that you would do whenever you get DNA, like ancient DNA. So for example, you sequence it and now you have the data. So this is the whole genome sequence. First, what you're going to do is you're going to plot it on a chart or on a PCA with every single worldwide population. So you see in this chart here, you have every single, basically just broad worldwide populations. And the Nueret individual is a big red circle. So you see blue is West Asia. So okay, modernly you can project them onto modern West Asian samples. So that now this gives us a starting point. And this is actually a very, very important step because you can then tell that if you like clustered all the way right here, then you know that it's highly contaminated or just something went wrong with the analysis process or with the extraction. Or if you clustered with Native Americans, it's either it was contaminated or this is the biggest discovery of the 21st century, which is not the case. So yeah. So once you have the modern populations, now you can narrow it down. So now we can curate all the ancient populations and we can kind of see which ancient populations he was closest to. So let's try to read this chart. We're looking at the red circle. So let's focus on the red circle. His closest populations are Levant Neolithic, and then the green triangle is Morocco Middle Neolithic. And he clusters in between. So this is just a good starting point. PCA is very noisy. It's very speculative, but this is a good starting point. So now we can do like a further analysis. And now we get to the admix analysis. And this is the fun stuff. So remember what I said, like how you can measure like different ancestral breakdowns. So actually we don't really care about all these. We just care about this one right here. But this is just directly from the paper. So over 70%, we can see that he derived it from a Moroccan Middle Neolithic ancestral source. And this was around the late Neolithic period, so after the Neolithic Revolution. It doesn't necessarily mean that this individual was of Neolithic Moroccan ancestry, but rather there was a widespread Neolithic Egyptian ancestry that just happened to be closest to this Neolithic Moroccan that we have. And I don't know if that made too much sense, but it's just the closest ancestry that we can pinpoint to this ancient Egyptian. So it was probably a shared common Neolithic source. And so they may have had a common North African ancestry. Yeah. And the rest of him is actually Mesopotamian. So you have that a Mesopotamian component, likely an Eastern Fertile Crescent related population. And this is kind of makes sense with the historical records. So ancient Mesopotamia, ancient Egypt, they had a lot of trade, population migrations. Both regions had farming, so the Neolithic centers of early farming. And yeah, so here we go. This is just on the map. So you see it's like just the different sources they're circled of where his ancestry may have come from. And again, like I said, it doesn't mean that he was exactly from these areas. It's just the closest ancestry that we can pin it to. That just happens to be in the DNA record. All right, so this is the Nueret individual. We see that he's likely Neolithic North African with a Mesopotamian component. So now let's look at the Fayum mummies, and these ones are really cool. So you had actually mtDNA from over 90 individuals, but remember, and I'm not going to say that we don't care about mtDNA, it's just we can't do as much cool things as we can with nuclear DNA. So we really care about the nuclear DNA, a whole genome sequencing from the 3 individuals, which are mentioned below. So they were from Abu Sir al-Melek, which is an archaeological site near Fayum, Egypt. It's on the map right there. And these samples were interesting because they showed a very, very high Levantine or Near Eastern ancestral component, which we're going to see in a bit. And I don't know if you guys have ever heard of Fayum. So Fayum, Egypt, you actually had the Fayum mummy portraits. So these were hundreds of years later, but these individuals actually may have looked very similar to this. So it was around like the similar necropolis or the place where they were buried. All right, so let's go ahead and plot them on the PCA. So you see these are our ancient Egyptian samples, and then we have our Near East, all our world populations. And we can see that they cluster closer to the Near Eastern samples, actually. So you got the yellows, you have like Levantine populations. The red, you have these Arabian, North African populations. So he clusters where we'd expect, or they cluster where we would expect them to cluster. And actually, this paper was done in 2017, so it's actually older than the previous. So that's why we didn't have really many ancient samples to compare it to. But the 2025 paper actually kind of compared them to the Nueret individuals. So we have the third intermediate period right here, and they're actually clustered pretty close to the Nueret individual previously below. So we can see a pretty common ancient Egyptian ancestry that just happens to cluster between Neolithic and Levantine Neolithic populations. And the paper didn't actually have an ancestral breakdown, so I just created this myself. So the mummies are a Levantine North African mixture. And again, like I said, it doesn't mean that they were exactly from these areas. These are just the closest population sources that we can pin it to that we have in the DNA record. So 45 plus or minus 5% Neolithic North African, 55% plus or minus 5% Bronze Age Levantine. And now I'm going to teach you guys how we can actually read these charts if you were to ever come across a DNA paper. So you see the p-value right here. The authors, they generated these values, and then they got a p-value, so 0.33. I think the general consensus, if it's below 0.05, then it's like you can accept the p-value. It's actually the opposite whenever you're doing the admixture analysis. So you want it to be above 0.05. So 0.33, it's a pretty good p-value. That means we can accept this model. So it doesn't mean this is 100% their ancestry, but it's just a model that we can accept because it's very hypothetical. It's a hypothesis. And okay, so another thing, I actually was wondering if the previous individual, he predated them by about 2,000, 1,500 years. Could we then model his ancestry as a Neolithic African source when using it instead of Neolithic North African? And this model was rejected. So we got 3.00 times 10 to the negative 7, which is way, way below 0.05. So for that reason, we can't actually say that there's genetic continuity, although it does mean that the ancient Egyptian population at the time was very diverse. So they likely had a common ancestor because we did see that the Neolithic North African was in the Nueret individual. So just an overall common Neolithic North African ancestor in ancient Egypt. Now, this study, though, did have some criticism. There's actually a massive Wikipedia article that just goes just bashing this article. You had a small sample size. You can't really say a lot when you only have 3 individuals for a 5,000-year historical time period or a 5,000-year-old historical culture. And then the Fayum actually only represents a small portion of the ancient Egyptian population. There were some overstatements in the article. And then again, Upper Egypt is going to naturally have a higher sub-Saharan ancestry, and then Lower Egypt is going to have lower sub-Saharan ancestry. And then remember, whenever you're talking about Egypt, it's actually in reverse. So Lower Egypt is actually north, Upper Egypt is south because the flow of the Nile, it's opposite. It does not go north to south. And then another thing was actually these samples were from the Fayum Oasis, and known through historical documents, it was an area where mercenaries of Middle Eastern ancestry were settled during the Late Kingdom. So for that reason, it's like there's some biases so we can't really take this like representative of the entire ancient Egyptian population. All right. And actually, I said in the few slides ago, I showed whenever I did the breakdown of all the ancient Egyptian samples, I showed you guys that there was one sample was discovered outside of ancient Egypt. So this was discovered in Lebanon. Not only did they find an ancient Egyptian, but they also found an ancient Egyptian and her son. And I'm going to show you guys how they found it. It's really fascinating. So this was around the Iron Age 2, so between 567 to 400 BC during the Persian period, so the Persian period of ancient Phoenicia. This was actually discovered in a multi-layer archaeological site in Beirut, Lebanon. So multi-layer, which I think we're gonna get to stratigraphy later. It's like you have different layers, meaning different probably time periods. So this was found in the Iron Age 2 layer. And this was actually an ancient Egyptian woman who was SFI 43 and her mixed half Levantine, half Egyptian son. And they were buried side by side. And the genomic profile of these two individuals are very similar to the Fayum mummies. And how did the scientist for this paper, Dr. Haber, how did he find these? Well, he plotted two populations. So he had the modern Lebanese populations and the modern Egyptian populations. And he noticed that one of them closer to the Egyptian populations, and one of them closer more towards the Levantine Lebanese populations. And then you have your ancient Egyptian samples. These are the Fayum mummies. So they were pretty close genetically when plotted on the PCA. And so now you can do the admixture proportions. So like we said, we can go ahead and calculate the different proportions. This is actually what an admixture proportion table looks like. So for example, you put in two test populations. So put in maybe population A, population B, it returns the p-value, and then it returns like the admixture proportions, that what it thinks, what the model thinks. So for example, it thinks in this one, A is 73%, so 73% from SFI 43, 27% from Beirut Iron Age, plus or minus 7%. So that's what the standard error means in this. So with this, we could see that SFI 44, the son, there's strong models that it's actually a mixture between that lady and a Levantine, ancient Levantine population. But how do we know it was her son actually? And with this, we can actually, this is very a lot. So what you're doing right here is you're estimating kinship by testing out different individual samples with each other. So for example, here it tests probably, you had about maybe 50 samples, you're testing each one against the other. So 50, I don't know how many pairs that would be. But whenever you test SFY 43 with SFI 44, it falls between the first degree individual lines. So for example, here you have second degree relationship, first degree relationship, identical twins. So these two samples, they went under the first degree relationship, meaning probably that they were siblings or a mother-son relation. That's a kinship. And those are actually all, so I showed you guys all the ancient Egyptian samples we have. So we see they're highly, in ancient times, they're highly Levantine. Of course, it's very biased. But now this raises the question, can we map genetic continuity into the modern era? And yes, we can. So interestingly, Schleinemann and all showed that we can with minor ancestral components because, I mean, in just history in general, you have a lot of population turnover, you have admixture events. Ancient Egypt saw the Greeks, the Romans, the Arabs, the Ottomans, the Turks. So it's really difficult to say like there's 100%, but there's probably minor components added with each different invasion or civilization. And again, it's difficult to determine due to the small sample sizes in the aDNA corps, but what we have, we can see. And another thing, so Egypt is very diverse. Most of the samples we have are from North Egypt, so it's going to be closer to the Mediterranean. So you're just going to have more trade, more population mixing. So for that reason, it's definitely a little biased towards Northern Egypt or Lower Egypt. Although Southern Egypt likely has a higher genetic affinity to the Sub-Sahara, so it's closer to Sudan. with the north showing affinity to the Levant. And this is actually a picture. I saw this trending. I think I saw it on Instagram, but it was trending on Reddit, on social media. So this is the guard of the tomb of Akhenaten, and this is the tomb that he's burying, that he's guarding. And everyone was like, "Oh my God, it's like Akhenaten's been reincarnated because they just look so similar." But yeah, there is genetic continuity between the two, between the modern era and ancient times. And again, here's a chart. So we see that whenever you do plot the ancient samples, they cluster more towards the Levantine samples, although they're pretty similar to ancient, pretty close to ancient Egypt or to the modern Egypt. And yeah, here we go. So modern Egyptians. And okay, so the ancient Egyptian race controversy. This is a really, really interesting one. So there was like this big debate back, I want to say it's been going on for over 100 years. And people were saying, oh, were the ancient Egyptians, were they sub-Saharan African? Were they white? Were they intermediate? And this was a big, big thing for many years. And now the modern scientific consensus is that ancient Egypt was, of course, a diverse indigenous civilization, and the populations varied by region and era. But overall, Egyptians belong to the MENA category, so Middle East, North African. But then on top of that, we can look at all these different civilizations or all these different dynasties. So, for example, we had the Ptolemaic Greeks. They were of Greek descent. So you had, for example, Ptolemy of Soter. They were Greek. Cleopatra was Greek. But then we also had the Nubian pharaohs of the 25th dynasty. So the Nubians likely resembled Sudanese. I think a lot of Sudanese they claim descent from the Nubians. So yeah, the Nubian dynasty. You had all sorts of dynasties. So here we go. We got Nubians. They're in modern day Sudan and South Egypt. So a higher sub-Saharan African component. So this is an S1. And then we have Egyptians today. So that's the thing. The moral of the story is that you cannot pin entire regions as comprised of a single ethnic group, but rather a continuum and mixture of different ethnicities.